Hello, and welcome to the FDA public webinar for the Patient-Focused Drug Development Draft Guidance 4, incorporating clinical outcome assessments into endpoints for regulatory decision-making. My name is Shannon Sparklin, and I am a member of the Patient-Focused Drug Development staff in the Center for Drug Evaluation and Research. I will be facilitating the webinar today. Next slide, please. Next slide, please. Thank you. The draft guidance has an associated public docket in which interested stakeholders are invited to submit comments on the draft guidance. The docket is open to the public and will close on July 5th, 2023. To submit a comment, please access the docket using the link on the slide and click comment. If possible, please submit your comments in a Word document format. When applicable, it is helpful to specify line numbers and proposed text revisions for comments and proposed changes to the guidance. In addition to any proposed changes, it is also helpful for us to hear what you like about the guidance so that we can consider both types of feedback when determining what to revise or remove. Next slide, please. Here's our agenda for today. This webinar will run from 1 to 3 p.m. Eastern Time. It will be recorded and posted online. The slides will be available and closed captioning is being provided. Now I will turn it over to Dr. Laura Lee Johnson to go over the agenda in more detail and to provide an overview of the PFDD guidance series.
Thank you, Shannon, and hello, everyone. As you can see, we have a full agenda today, hearing from several speakers on topics related to the draft guidance. Unfortunately, we don't have time to discuss all the topics and guidance for today, nor will we be able to cover all the complexity of each topic we do discuss. We encourage you to put your questions in the chat and we'll address them as time permits at the end of today's webinar, but we will read all of them. Next slide, please. The PFDD guidance series builds on the foundations of the 2009 guidance, and it's necessarily different. Next slide. The methodologic guidance series as a whole, we have to address all types of COAs, and we have a lot of feedback on the original 2009 PIRO guidance and from the public meetings over the years. Next slide. Remember, in developing this methodologic guidance series, we had public workshops prior to issuing the draft guidance. These meetings were part of our process to help us create draft guidance. I emphasize the path and evolution because some are concerned that the third and fourth draft guidances in the series look different than the discussion documents. The discussion documents for public workshops are not draft guidances. They're discussion documents for the public meetings, and we had and heard a lot of discussion at the workshops and also in the dockets. We heard from several other sources of feedback as well. We heard from internal communities. We also looked at how the comments we were giving to sponsors were going and what the responses were that we were receiving. Also, we had several different discussions with patients groups, and all of these were evolving. Our internal discussions to come to the draft guidance were a combination of the scientific discourse with experts, FDA experiences with feedback that we're commonly providing, and the stakeholder input. This includes meetings FDA and others host and programs funded by international stakeholders such as Europe's Innovative Health Initiative and literature published in recent years. In some cases, we realized the information in the discussion documents had not evolved as much as was needed from the 2009 guidance. To accommodate the science that grew inside and outside the regulatory setting and the expansion to all types of COAs, we needed to evolve even further in our writing. We understand many times guidances tell you what's been done in the past. This series has the past in mind and is very future focused. Next slide. This leads to what you see in the draft guidances in this series, especially guidances 3 and 4. And then the process in part happens again. The drafts are circulated for public input with a docket for comments, which is where we are today, and continued internal FDA discussions and listening. While draft guidances are not for implementation, some groups will implement, kick the tires, so to speak, And so we all learn from those experiences, and we take those learnings and all the information we've gathered to inform our revisions, which will lead to finalization. Next slide. As we move to final guidance, and all along the way, there'll be trainings internally and externally and public workshops. Next slide, please. The framework for arriving at COAs that are fit for purpose and to construct the endpoints in clinical trials is provided by the PFDD guidance series. This series of 4 guidances describes how stakeholders, that's patients, caregivers, trialists, medical product developers, researchers, and others, can collect and submit patient experience data and all other relevant information from patients, caregivers to be used for medical product development and regulatory decision-making. Note that guidances 1 and 2 are finalized, and guidances 3 and 4 are currently published as draft guidances. Now let's look at how the guidances work together and build on each other. Next slide, please. Guidance 1 discusses understanding the disease or condition from the patient's perspective. It provides recommended methods to collect patient experience data that are accurate and representative of the intended patient population. This guidance helps to ensure that systematic approaches are used to determine the research design of patient experience studies. Next slide. Guidance 2 discusses how to conceptualize clinical benefits and risks. It provides recommended approaches to identify what is most important to patients' experience with the burden of disease or condition and the burden of treatment. This guidance shows how different research methods can be employed to collect robust, meaningful information and what aspects of symptoms, disease, impacts, and other issues are important to patients. Next slide. Guidance 3 discusses how to select or develop clinical outcome assessments, or COAs, that are fit for purpose. Next slide. The guidance outlines 3 paths for doing this. Sponsors may search for an existing COA that measures the concept of interest in the context of use. They may find that a COA already exists for that, and therefore the identified COA can be used unmodified. They may find that a COA exists for the concept of interest, but it's not yet been validated in the context of use. In this case, the sponsor would have to collect evidence and modify the COA as necessary before it can be considered fit for purpose. Finally, you might find that there's no existing COA that's measuring the concept of interest. In this case, the sponsor may decide to develop a new COA or empirically evaluate its performance in order to gather evidence to show the COA is fit for purpose. In each of these situations, a sponsor has to provide a rationale for why the COA is fit for purpose in their context of use, and they must support the rationale with evidence, either from the literature or from their own data collection and analysis. Next slide. Once patient input has been used to understand the disease or condition and to conceptualize the clinical benefits and/or the risks, and a COA has been shown to be fit for purpose within the context of use, the COA can be used to construct an endpoint. Guidance 4, the focus of today's webinar, discusses how to incorporate COAs into endpoints for regulatory decision-making. This includes construction of the endpoint, assessing the endpoint in a clinical trial, and analyzing and interpreting the meaningfulness of treatment effect. While we will not cover all of Guidance 4 today, we did want to touch on a few of the topics that may be of most interest, since they may be new or fundamental to our discussions. We'll also see reference to many other guidances that are relevant to endpoints that Guidance 4 interweaves with, such as the multiple endpoints guidance that was finalized in 2022. You might realize that and see this intentional shift in nomenclature in the guidance series. Many discussions over the last few decades have started with, this is the language so-and-so used. So we took a step back, and FDA reviewers and decision makers, sponsors, we have a specific task looking at data coming from clinical investigations. We're trying to interpret treatment effects. So instead of trying to backfit, we said, how do you do this task? The words and terms that we're using are telling the reader the task that we are attempting to do so we can have the data and analyses to accomplish the task. 2 more quick notes. We were asked if we were walking back from within patient change, and many of us need to work both within the patient and also draw inference comparing patients who received an interventional product to people in a control group. Both types of information come into FDA. We expect sponsors to be clear if the values they're talking about are within patient differences or differences between groups. They're pretty different, no pun intended, and it's confused a lot in the literature. We tried in the guidance to put out an overall set of mappings and frameworks to be flexible for a lot of different needs and accommodate broader ranges of methods. We also want to be clear. Today, you're going to hear from FDAers who are clinical signatories, statisticians, a COA director, psychometricians. We all work together both to use and in developing guidance for and expect externally that the same will occur. This is not a single discipline or therapeutic areas guidance. So the core principles are still the same. Good science is good science. And in this part of this process, we are expanding the frame. This series of guidances will go past the 2009 PRO guidance, and there are changes because we're providing changed comments to our sponsors and patient groups today. Also, and finally, this is a guidance, it's not a textbook. The guidance provides recommendations that encourage rigor and flexible approaches. Shannon, back to you.
Thank you, Dr. Johnson, for providing that introduction. Moving on, we will now hear from Dr. Lily Gerard from the Division of Biometrics 3 in the Office of Biostatistics in the Center for Drug Evaluation and Research. Dr. Gerard was unable to join us live today, but we very much appreciate her recording her presentation.
Hello, I'm Dr. Lily Gerard. I'm a master scientist and the technical lead of patient-focused statistical support in Division of Biometrics 3, CDER Office of Biostatistics. I will focus my discussions on considerations for COA-based endpoints, which is an important topic area in the draft guidance 4. Before getting into the specifics, let us review the purpose of a COA-based endpoint. Generally, endpoints constructed from fit-for-purpose COA measures should reflect an aspect of the patient's health that is meaningful and be capable of supporting an inference of treatment effect within the context of the planned clinical trial, which is a fancy way of saying you're able to use it in the statistical analysis of the clinical trial and make sense of everything in the end. When defining a COA-based endpoint, sponsors should clearly describe the endpoint in terms of the following: the type of COA assessment made, for example, a patient-reported outcome measure, the COA used to measure the concept of interest, the specific score from the COA, for example, a total score computed from all items on a COA, a clear definition of baseline if applicable. Is it a single measurement on day 0 before people start the intervention? Are you averaging 14 days of daily diary responses before they start? Or is it something else? What is the baseline measurement? The timing of assessments, time frame over which COA scores are combined, and how COA scores are combined into an endpoint. The rules for handling missing item responses or task results for scoring, along with the justification for the rules. If a multi-component endpoint is proposed, specify the algorithm used to combine scores from different components into a single endpoint. Now we're going to go through 3 COA-based endpoint considerations. FDA recognizes that constructing and selecting trial endpoints often involves weighing the strength and limitations of different approaches. The first point of consideration and a key message is that sponsors should provide a well-justified rationale for the choice of endpoint early in the planning of a clinical trial. This complements Draft Guidance 3, which discussed the need for a compelling evidence-based rationale for interpreting COA scores as measures of the concept of interest. Draft Guidance 4 suggests several things to include in the rationale for the choice of endpoint: the concept of interest, the clinical trial objective or hypotheses corresponding to the endpoint, the role of the endpoint, for example, primary, secondary, or exploratory, intended indication related to the COA-based endpoint, explanation for why the selected COA is fit for purpose in the planned trial, support for importance of endpoint to patients and/or caregivers. For a multi-component endpoint, justification for the individual components included and the algorithm for combining them into an endpoint, strengths and limitations of the proposed endpoint. Having a well-justified rationale not only helps sponsors facilitate clear communication with FDA, but also helps facilitate more efficient review and decision-making by FDA. Now let's think about time. An analysis at a fixed time point might be justified if the COA score is not highly variable over time, and the chosen time point, for example, end of study, would be useful for reflecting the durability of the treatment effect. Justification of the fixed time point should also take into consideration the recall period of the COA. Now, if an endpoint is defined based on COA scores summarized over time, it should be recognized that different summaries may be appropriate. You will hear some examples about different ways to summarize data and how to select from those endpoints in the next session from Arthur Stone. Sponsors should provide justification for their chosen summary, and the decision process should consider the robustness of the summary or model and any modeling assumptions. For some types of summaries, an alternative approach is to use repeated measures modeling of all observed COA scores and derive summary estimates from the model. You should consider handling of missing COA scores, statistical power, and interpretability of the endpoint. A second point of consideration is that endpoints defined as COA scores at a fixed follow-up time, analyzed by conditioning on baseline COA score, are generally preferred over endpoints defined in terms of responder status, change from baseline scores, or percent change from baseline scores. Draft Guidance 4 states, in most situations in which a COA produces ordinal or continuous, that is interval or ratio scale scores, the best and recommended endpoint will be the COA score at a predefined assessment point or summarized over some predefined post-baseline assessment period. The guidance goes on to say, and the most straightforward analysis will be a comparison of randomized groups with respect to the follow-up scores after adjusting for the baseline value, for example, with a linear model to compare average follow-up scores. Now, what about the other commonly used COA-based endpoints, defined in terms of responder status, change from baseline scores, or percent change from baseline scores? The Draft Guidance 4 describes specific limitations and considerations for each of these approaches. Next, I will discuss each approach in more detail. An endpoint defined using a responder status, or commonly referred to as a responder endpoint, is constructed by dichotomizing ordinal or continuous COA scores. Defining an endpoint based on dichotomization may use less information and reduce statistical power. However, in some situations, such endpoints may be reasonable when evaluating the effect of a treatment on the probability of achieving a clearly defined and important health state, such as complete symptom resolution. With that said, if a responder endpoint is considered, it is important to pre-specify a single score threshold that is used to define the responder status and provide compelling evidence to justify the dichotomization. For example, the justification could be supported by strong qualitative evidence that patients and/or caregivers view the health states above the responder threshold to be meaningfully different from the health states below the responder threshold. Another critical consideration for using a responder endpoint is that data used to derive a score threshold should differ from data used to demonstrate effectiveness. While we see many endpoints defined in terms of change from baseline scores, this approach has limitations. It may be challenging to interpret a change from baseline endpoint for ordinal COA scores, as the difference between two ordinal scores cannot be assumed to have the same meaning across scores. Or in other words, moving from mild to moderate may not have the same importance to a patient as moving from severe to very severe. Mathematically, many times they do. We have to remember there might not be a linear relationship between the ordinal values and the true level of symptom severity or functioning being measured. As previously mentioned, in comparative trials, the preferred method for adjusting for baseline status is to do so in the context of a statistical model, for example, as a covariate in a linear model. But note that for situations in which a single-arm trial is the only viable option, we acknowledge that a change from baseline endpoint might be the best available option. A similar endpoint is defined in terms of percent change from baseline scores. The interpretation can be complicated because of the asymmetric nature of the endpoint, which treats baseline and follow-up COA scores differently. Let's go through a brief example. This patient's COA score improves from 5 to 10, a change of +5. The patient has 100% improvement, which is wonderful. Now this patient's score decreases from 10 to 5, a change of -5. The percent change is a 50% decrease, maybe not so good. It's clear that 50% is not 100%, but both patients move between 5 and 10. This has important implications, including the fact that the average change on the original scale, which is 0, indicates no overall change, whereas the average percent change is a positive 25%, suggesting an overall improvement. Additionally, the percent change from baseline is undefined if the baseline COA score is 0. Such change scores may have highly non-normal distributions that can be challenging to model. Finally, as an alternative, if the effect of the treatment is expected to be multiplicative rather than additive, then a log or similar transformation could be applied to continuously distributed COA scores. But we know that transformation could complicate interpretation. Now that I've discussed considerations and limitations for these other commonly used endpoints, when should we actually use them? Sometimes people consider using one of these 3 endpoints because they think the results might be easier to understand or communicate to some stakeholders. If this is the case, we can still estimate the treatment effect using a model for ordinal or continuous COA scores, and then communicate the results of the model in terms that might be easier to understand, such as thresholds, change from baseline, or percent change from baseline. We will move on to one of the major challenges to COA-based endpoint construction, the heterogeneity in diseases that all stakeholders often have to deal with. First of all, it should be stated that there is no perfect endpoint strategy when a disease affects multiple aspects of feeling and functioning between patients and within the same patient over time. Because of this, it may be necessary to consider several different aspects of health to adequately assess benefit. For instance, some aspects of health might be relevant for almost all patients with a given condition. Other affected aspects of health might differ between patients and within patients over time with certain conditions. In these situations, it may be challenging to identify one specific aspect of the disease for evaluating treatment benefit. Sponsors should consider the strength and limitations of various approaches, and when possible, evaluate multiple endpoints in earlier studies to inform endpoint selection for later studies. Note that draft guidance force discussion on this point is complementary to the multiple endpoints guidance finalized in 2022. With that said, the third and final point of consideration is that there is no perfect endpoint strategy when a disease affects multiple aspects of feeling and functioning, so sponsors should choose the best for their context of use. Draft Guidance 4 discusses 3 main strategies to use when a disease affects multiple aspects of feeling and functioning. The first is to construct separate endpoints for each aspect of health. The second is to construct a multi-component endpoint. And the third is to construct a personalized endpoint. The guidance discusses pros and cons for each of these strategies in detail. We will use an illustrative example to explain the 3 general strategies. Suppose we have 5 different outcomes of interest with a disease. The first strategy is to construct separate endpoints for each aspect of health. In our illustrative example, we will have 5 separate endpoints corresponding to the 5 outcomes of interest. With this approach, sponsors should describe the role of each endpoint. By creating a separate endpoint for each relevant aspect of health, there's clarity about which aspect of health has or has not been affected by the medical product. Because each endpoint corresponds to only one aspect of health, but there are several challenges with this approach that also should be considered. First, for diseases with many possible manifestations, the approach may be challenging to use if it is not known ahead of time which aspects of health are most likely to improve as a result of using the medical product under study. Second, depending upon the roles of the endpoints, multiplicity adjustments might be needed, necessitating a larger sample size to ensure sufficient statistical power. Finally, if patients differ from one another in their symptom or functioning due to the disease, then the treatment effect estimated for any one endpoint will be diluted by the patients for whom the endpoint is not relevant. Example, patients who never had a given symptom cannot improve with treatment. Next, we will move on to the second general strategy, which is to construct a multi-component endpoint, defined as a within-patient combination of 2 or more components, each reflecting a different aspect of health. Constructing a multi-component endpoint for an individual patient requires observation of all the specified components for that patient. Then a single overall rating or status on the endpoint is determined according to a pre-specified algorithm. A COA-based multi-component endpoint may take many forms. Option 1 is where each component of the multi-component endpoint could be the score from a different COA. In this example on the right, suppose we have 5 different COAs, each evaluating a different symptom severity, ranging from symptom A to symptom E. Using a pre-specified algorithm and either implicit or explicit weights, we can combine the components into an endpoint. Option 2 is where each component could be the score from a subdomain of a single multidimensional COA. For example, suppose we have a multidimensional PRO measure called ABC functioning measure that consists of 5 subdomains. Each subdomain might include several items or tasks. Again, using a pre-specified algorithm, we can combine the scores from the 5 subdomains into an endpoint. Option 3 is where each component could be the response to an item or task from a single COA. Suppose we have a COA that consists of 5 different tasks, task A through task E. An endpoint evaluating the disease X functioning score at week 12 can be created from responses to the 5 tasks. Of note, a unique consideration for option 3 is that option 3 should only be used when the COA is based on a composite indicator model. A composite indicator model is a specific type of measurement model for COA development, and details are discussed in Draft Guidance 3. Under the composite indicator model, responses to the items or tasks are not assumed to be reflective of or caused by a single underlying aspect of health. Each item or task addresses a separate health concept, and when combined, responses to all the items or tasks define the overall concept of interest. Here's an example composite indicator model for a COA, where the concept of interest, activities of daily living, might be defined as the degree to which the patient is able to accomplish everyday tasks that are necessary to live independently. This slide illustrates how responses to the 5 tasks included on a single PRO measure, developed based on a composite indicator model, can be used to construct a multi-component endpoint. Now we have seen the different ways to construct a COA-based multi-component endpoint. Again, it's important to take a look at advantages and limitations. A COA-based multi-component endpoint has the potential to evaluate the entire range of important disease manifestations. No multiplicity adjustment is needed and can be efficient if the treatment effect on the different components are generally concordant. But again, based on practice and experience, we know it is not simple to create a multi-component endpoint. Creating a reliable and reasonable scoring algorithm can be difficult. And there are also concerns and limitations for different types of multi-component endpoints. There are many issues we have seen at FDA, which we put in draft guidance for. Some of them are related to the endpoint, and some are related to the algorithm. I want to transition now to the final strategy, personalized endpoint. Personalized endpoints are sometimes proposed to reflect what is important to each individual patient enrolled in a clinical trial. To construct a personalized endpoint, we again need to weigh the advantages against concerns. The advantages are, as the name suggests, this type of endpoint is very patient-focused. Aspects of health that are not a problem for patient are not included in the endpoint, meaning no dilution of treatment effect. A personalized endpoint could be considered along with another endpoint to inform decisions about the effect of a medical product. An example personalized endpoint for a specific context of use is described in FDA guidance for industry, migraine, developing drugs for acute treatment, published in 2018. And the concerns are, if an endpoint is based on, for example, most bothersome or most severe symptoms, it might be hard for patient to pick a single symptom. In addition, the most bothersome may or may not be the most severe for different patients. A patient's view of most bothersome or most severe symptom might change during the duration of a trial. If goal attainment scaling, or GAS, is used to define a personalized endpoint, patients might choose symptom or functioning that is unlikely to be affected by the medical product during the trial, or the chosen goal might change during the trial. Lastly, a few reminders. When constructing and operationalizing a personalized endpoint, we should assess the same set of outcome measures for all patients, regardless of their own personalized endpoint, to account for any new or worsening symptoms and or functioning limitations occurred during the trial. We need standardized process for eliciting personalized endpoints, including standardized criteria to select outcome measures. And lastly, in addition to symptom or functioning identified as the most important to the individual patient, it is important to measure all relevant symptoms and areas of functioning to support any additional analyses of treatment benefit. With that, I conclude my presentation today. The take-home message is that regardless of the choice of the endpoint, it needs to be supported by a well-justified rationale. Input from different stakeholders should be considered, especially input from patients and our caregivers. And early communication with FDA is highly encouraged. Thank you very much.
Again, we thank Dr. Gerard for taking the time to provide us with her presentation. As Dr. Gerard just mentioned, the endpoint rationale should include support for the importance of the endpoint to patients. With that being said, Dr. Arthur Stone, Professor of Psychology, Economics, and Public Policy, and Director of the University of Southern California Dornsife Center for Self-Report Science, will now present a study conducted to understand patients' and other stakeholders' views on a set of endpoints. Dr. Stone, please turn on your camera and begin your presentation.
Good afternoon. I'm Arthur Stone from the University of Southern California. Next slide. I appreciate the FDA's invitation to speak about the guidance today. And the study that I will describe, though, is or was done before the guidance was issued. And it really is a very small step that is consistent with the recommendations that you are hearing about today. Let me say that I fully recognize that the methods that I describe here are one of many different ways to approach the issue of bringing the patient perspective into outcome development. Before I get into the substance, let me mention the investigative team and the study citations. Next slide. The primary information that I'll be presenting today is based on the study mentioned or shown on the slide in blue. It's a series of 3 studies that were published in the Journal of Pain in 2021. The other 2 studies get into more depth about the kind of outcomes that I'll be talking about today. One gets into the psychometrics of it. The other gets into how it performed in a clinical trial. Next slide. So let me provide you some context for what we were doing. It was part of the development of new outcomes for assessing pain that were based on momentary assessments or ecological momentary assessment of pain intensity. The current view in the field is that the average pain intensity or the worst pain experienced for a designated period are what is being used. I think worst pain is actually getting more play these days than average pain. The measures could be based on diaries, or they could be based on 1-week retrospection, for example. The goal that we had was to use granular, repeated measures of pain intensity over a 1-week period. That is, measurements that were taken many times a day over the 1-week period, asking about pain intensity. And we were trying to characterize patients' pain experiences in new ways, ultimately with the hope of being able to understand facets of the pain experience that might improve medical treatment. The proposed outcomes were based in part on prior literature. For example, we know that pain variability is associated with poor functioning. It was also based on knowledge about what is missed with 1-week retrospection. For example, it's very difficult for patients to recall the amount of time that they weren't in pain. And we know that that's a cognitive heuristic known as duration neglect. Next slide. Just to highlight this, the reason for using these momentary assessments, this is a description of one patient in one of our studies. The x-axis has the day in the study. You can see there were many days. And the y-axis is a visual analog scale, 0 to 100, for pain intensity. The darkened triangles represent the average level of pain based on the momentary reports over the course of the study, day by day. The little dots represent each of the momentary assessments of pain. And the point here is that there's lots of variability, both day-to-day variability and within-day variability. And the methods that we are using are trying to capitalize and understand what's going on with this variability. Chronic pain patients don't just have one level of pain. It varies a lot. Next slide. So as I mentioned already, average or worst pain symptom levels are often what are used as outcomes. But is this what actually makes a difference to patients? Does it provide us with the optimal information that we want about a treatment effect? So alternatives to the average and worst have rarely been examined, so there's little information about the potential utility of these alternative conceptualizations of outcomes. So our objective was to explore other ways of characterizing the experience of pain over the course of a week based from these momentary reports. Next slide. What I want to do very quickly is simply go through some of the indices that we use. So we took the average of pain intensity. We also found when pain was worse based on the momentary reports. We found the level of pain intensity when it was at its least during a week. We found the amount of time the patient spent with no or low pain. Next. The amount of time a patient spent with high pain during the week. And then we also looked at how much pain intensity varied over the course of the week and also the amount of unpredictability that patients had in knowing when pain would occur. Next slide. As we were putting this research program together, it quickly came to light and it brought into very sharp focus the question about how would one know which of these trial outcomes should be used. And we could select one based on the literature or based on prior work, but that didn't seem the best way to go. So we tried to address the question of how do various stakeholders view the importance of these new pain indices that I just described. So let me tell you about how we did this study. Next slide. We took 3 samples of stakeholders. We took patients with chronic pain conditions, and you can see the details on the slide. We took 20 healthcare providers, again, with special training in pain medicine, and we took 20 clinical trialists, and these folks also had experience with pain. Understandably, although we wanted to chat with regulators, they were reluctant to engage with us in this task. Next slide. What we did was we conducted interviews with all of the stakeholders I just mentioned by telephone for about 20 minutes. Now, prior to telephoning people, we mailed them with index cards that had the definitions of the pain, the new pain indices that we wanted to discuss with them. We probed for comprehension. We had them read through them with us, and then we made sure that they understood them. Now, the important part of what we did here for today's talk is that we had all of the stakeholders rate the importance of each of the new indices, pain indices. For patients, we asked them to rate the importance of what they were most hoping to achieve from their treatment. For the others, for the healthcare providers and the trialists, we asked them to rate the importance for evaluating medical treatment outcome. And this was done with a card sorting task that's described in the paper, where rankings were 1 was most important, 7 was least important. We also tape recorded these interviews and have verbatim text, which gave us greater insight, but I won't be speaking of that today. Next slide. The patient characteristics, the clinician characteristics, and the trialist, you can quickly take a look at these. I will be the first to admit that they were not ideal, and I'll come back to that a little bit later. Next slide. So here are the results of this sorting task for importance. Let me take you through how to read this. So on the y-axis, we have the results of the sorting where 1 is most important and 7 is least important. Each of the symbols in the graphic are described in the key to the right. The oval circles surrounding the little graphics, the little symbols, indicate that they were not significantly different. The ratings were not significantly different. So for the patients who are presented here, worst pain, the ratings of worst pain and time in high pain were rated as the things that patients most wanted to change. Going to the opposite end, time in no pain and time in least pain were things that they were less interested in changing, whereas the other indicators, the other new outcomes, were rated somewhere in between. Interesting pain variability was something that patients rated as important. So now let's turn and contrast these results to the results from the healthcare providers. Next slide. Here, reading the graphic the same way, we see a very different patterning of importance, where clinicians were saying the thing that was most important for them to change in their patients with their treatments was the worst pain, whereas all of the other pain indicators sort of lumped together and were really not all that different from one another. So that's fairly interesting that the patients and the clinicians have a somewhat discrepant view. I mean, at least in terms of time and high pain. Next slide. And we turn to the trialists. The trialists tended to focus on average pain as being the thing that was most important. And this may go back to historical ways of doing things that maybe the trialists are still concerned about. Next slide. So let me comment on this. We were gratified that the new momentary-based pain indices were understood by stakeholders. And there were clear and differing opinions expressed by the stakeholder groups, which I've already mentioned, but worse pain was preferred by clinicians and patients also thought the duration in high pain was important. Clinicians didn't think that. And variability was also important to patients, giving us a hint about what we might be looking to change in patients. Next slide. Of course, this approach could be improved. We could have a much better representation of stakeholder populations. We could have increased sample size to allow for subgroup analyses, which might provide insights as to how these ratings are being made and for whom, which of the indices are most important. And we would have liked to have done these face-to-face, but we had to do them through remote interviews because these are simply more scalable. Now, some conceptual considerations for these results. If we decided that treatments should be directed at the patient-chosen outcomes, will the patients really be better off? It seems to me that this is a good start, but there actually has to be empirical work to justify that kind of decision, which would include all sorts of aspects of functioning that we might include in being better off. And certainly patient preferences are one aspect, an important one of a multifaceted approach, which is consistent with what my first point was. Next slide. So I believe these data show the importance of considering the opinions of multiple stakeholders, especially patients, for selecting outcome measures. They highlight the importance of various ways that pain intensity can be summarized when granular data is available, or are available. This is particularly important because what we do want to change is the patient's experience of pain and their functioning. I think the results also highlight the need for an empirical, systematic, and transparent approach to the development and selection of outcomes. Thank you very much for your attention, and I'd now like to turn the presentation over to Dr. Liu.
Okay, so can you hear me now?
Yes, we can hear you.
Okay, sorry about it. So after constructing suitable COA-based endpoints, statistical analysis considerations are in principle similar to other endpoints. I will cover briefly some statistical considerations that commonly arise in the analysis of COA-based endpoints. Next slide. Specifically, I will cover these three topics: analysis at a fixed time point, analyzing ordinal data, and missing data. Next slide, please. So for analysis, a fixed time point, when baseline COA scores are available, it's generally more statistically efficient in terms of increased statistical power to adjust for these baseline COA scores. This also applies when the endpoint is the change in COA score from baseline to a predefined time point. FDA's 2021 draft guidance of adjusting for covariates in randomized clinical trials has more information on this. Next slide, please. Next slide, please. So if COA-based endpoint is collected repeatedly data from intermediate time points can still be included in the longitudinal model in which a treatment contrast is made for a pre-specified fixed time point. Next slide, please. And sometimes CLA scores are used to construct an endpoint that results in an ordinal metric. Several analytical options exist for ordinally scaled endpoints. The choice of analytical approach might depend on the type of ordinal endpoints. In the guidance, we discuss two types of ordinal endpoints, those reflecting just one aspect of health, or those reflect multiple aspects of health. Sorry, we should move to the next slide. Yeah. Okay, so I'm talking about ordinal data. Next slide, please. So some ordinal endpoints is based on a CLA measuring only a single aspect of health. The guidance discuss some analytical approaches and potential issues. So for example, one approach is to model mean difference. For example, using ANCOVA, this approach can be difficult to interpret when steps between levels do not reflect equal increments. And another possibility is to dichotomize the endpoint. This approach can risk missing important information. And another option is to use ordinal models. So models designed specifically for ordinal endpoints, such as cumulative logistic regression. Regardless of the approach taken, sponsors should explore the potential impact of violation of assumptions. Next slide, please. So the second type of ordinal endpoints is the multi-component endpoints that's constructed by assigning ordinal values based on scores reflecting multiple aspects of health. For example, an endpoint may combine symptom levels, hospitalization, and death. While the same analytical approach could be considered for this type of ordinal endpoints, greater caution is required in interpreting the findings. So one concern can be treatment could be beneficial for one aspect of health, but harmful in terms of another. Sponsors should consult with FDA when developing analytical plans for such endpoints. Next slide, please. So the final topic here is the missing data. Next slide, please. Data may be missed only in some items in the CLA instrument, or can be missed entirely at some time points. And the approach can be to handle this can be different. Next slide, please. Next slide, please. Okay, so missing data may lead to reduced power and potential bias. So we should make every effort to avoid missing CLA data and to manage the impact of missing data. The guidance listed some steps that can be taken. For example, we should collect only the CLAs needed to assess endpoint, and we should make data collection plan easy and low burden for the participants. And we should counsel respondents on the importance of completing the CLA and provide timely reminders when CLA are need to be completed. And when CLA does come missing, sites should be notified so that research staff can adjust that timely. And we should also collect reasons for missingness. Next slide, please. So when only some items are missing in the instrument, sometimes it's possible to handle that based on the scoring algorithm of the instrument. Next slide, please. So when considering intermittently missing CLA, sponsors should propose statistical methods that properly account for missing data with respect to particular estimate that answer specific clinical questions. That's all for the statistical considerations for analyzing CLA endpoints. I will now pass the podium to the next speaker. Thank you.
Thank you, Dr. Luo. We will now hear from Dr. Hilton Jaffe. Dr. Jaffe was also unable to join us live, but he has provided a recorded presentation.
Good afternoon. My name is Hilton Jaffe, and I'm the Director of the Office of Cardiology, Hematology, Endocrinology, and Nephrology within the Office of New Drugs in CDER. And I'll be giving you the clinical perspective of incorporating clinical outcome assessments into endpoints for regulatory decision making. As I mentioned, I'm an office director, and previously I was a division director, all within FDA's Office of New Drugs. And I've been the signatory authority for certain drugs and biologics in these roles. Over the years, I've seen the use of clinical outcome assessments, or COAs, across a variety of diseases, including cardiovascular diseases, non-malignant hematology, endocrinology, rare diseases, urology, and gynecology. And what I'd like to do is share some common issues I've seen with COA measures. And some of these issues are covered in our PFDD guidance number 3, as well as common issues with COA-based endpoints when they're used in trials for establishing drug effectiveness. Now, of course, I won't be able to cover all the clinical aspects of COAs as trial endpoints in this 10-minute talk. So I encourage and recommend all of you to spend more time with our 4 PFDD guidances, which incorporate principles from FDA's wide-ranging experience in drug development over the years. What I'd first like to turn to is suboptimal instruments. And when you use a suboptimal instrument as an endpoint in a trial, what happens is that suboptimal instrument adds noise to the measurements, and in a superiority trial is going to bias results towards the null, meaning towards showing no effect of the drug. And so a suboptimal instrument actually makes it harder to show a drug effect even when there really is one. And so this can lead to an underestimation of the drug's true effect. And when that happens, you could end up with a very small treatment effect, for example, and that could lead us to question whether that result is clinically meaningful. Also, it could impact benefit risk assessments, because if you have what appears to be less benefit on the benefit side of the equation, and you have serious risks, that benefit risk assessment could turn unfavorable. Now, also, suboptimal instrument can also lead to uncertainty about what the instrument is actually assessing as well. Now, in a non-inferiority trial, when you add noise with a suboptimal instrument, that could actually bias results towards showing non-inferiority, even if a drug was actually inferior to the comparator. So we really want to avoid using suboptimal instruments in our clinical trials. This slide here gives you some examples of suboptimal instruments that I've seen over the years. It's not an all-inclusive list, but things that I've seen include things such as vague or confusing or ambiguous questions in the COA instrument that's going to be used for the endpoint. And oftentimes these can be seen just on face value. I've also seen instruments that try and assess distal concepts. So an example would be, say you're having a trial that's assessing a pain medication and you're looking at a pain symptom, and then want to look at, say, quality of life. Well, quality of life is quite distal to pain. Of course, pain could impact quality of life, but there's many other things that could impact quality of life as well, including a patient's comorbidities, disabilities, and so forth. And also different patients may interpret quality of life differently. They may think about what that means differently. And so when you use something like a very distal concept, what happens is that yes, the drug might impact some aspect of the quality of life, but there are other things that could be impacting the quality of life measure as well. And that just makes it harder to really tease out the part that is coming from the drug. Sometimes I see companies try and measure a broad concept, for example, cognition, with a single item. And broad concepts like these need more items to really get at what you're trying to measure. I've seen instruments that include concepts that are not relevant to the disease being studied in the trial, or also concepts that aren't sensitive to change with the drug. So really think carefully about what the mechanism of the drug is, what you expect the drug to be able to change, and then make sure your endpoint really is trying to capture that aspect of the drug. Of course, that aspect still needs to be important for patients. Other things I've seen are what we call multi-barreled items. So that's where you you have an item in an endpoint, and that item contains more than one question. And so it's very difficult, impossible really, to tell if there's improvement in that item, whether it was improvement to one of the questions, both of the questions, etc. And then sometimes I see response options that can be hard to tell apart. Asking a patient, for example, to choose between a slight symptom versus a mild symptom. Turning to recall periods, this is another area where we often see recall periods that are too long. A recall period is basically where you're asking a patient to reflect back and recall how they felt, for example, about a symptom that they're reporting on for the endpoint. And you want to make sure the recall period is the right length of time so that patients can validly recall the requested information. When you have a recall period that's too long, that typically adds noise. It's hard for patients to remember accurately what they felt like, for example, 4 weeks ago, if you're asking about pain from weeks ago. Also, when you have very long recall periods, usually what patients end up doing is being more heavily influenced by their recent experiences. So think about what the most appropriate recall period, and realize in some situations you might even need shorter recalls than you were anticipating. For example, if you have a disease that affects cognition and you're enrolling patients with significant cognitive impairment in your trial, you can't have a long period because of the patient's disease state and their ability to remember. Another issue I see not infrequently is what I call instrument burden. And this is where you have overly burdensome instruments that you're asking patients to fill out. And this could be either that the instrument that you're using for your endpoint is very long, has a lot of items to it, or you might have a bunch of other instruments that you're asking patients to fill out also, or you might even be having patients fill out these instruments that aren't in themselves very burdensome, but you're having them do it very frequently. And all of these things can lead to what is called diary fatigue, where patients basically get tired of repeatedly filling out all this information, and that can lead to missing data for your COA endpoint, which is really something we want to avoid. Also, patients having to continuously fill out these instruments can lead them to them picking out the same answer again and again. So really think very carefully to, what are the real critical items for the instrument that you're using for your endpoint? How frequently do you really need to administer that instrument? And then think about if there's other trial burdens that you may be able to minimize. So for example, if you are also doing a slew of other instruments, but they're for exploratory endpoints, really think about how critical that is for your trial. Another concept is floor effects. And what happens here basically is that a floor effect occurs when a high percentage of subjects in your trial select the least severe response for an item. And the problem here is that when subjects select the least severe response, they don't really have any room for improving on that item, even if the drug's effective. And so think carefully about this issue when you enroll patients in your trials. Make sure they have sufficient severity of whatever symptom or other measure you're using on your endpoint, and make sure they have sufficient severity at baseline so they have room to improve. So pay attention to your inclusion-exclusion criteria. Use the same instrument at baseline that you'll use for the endpoint. Make sure patients meet a certain level of severity on that instrument at baseline. Blinding, of course, blinding is a very good tool that we have in general in terms of minimizing bias. It's particularly critical when you have a COA-based endpoint, because we're always worried that when a subject knows their treatment assignment, it can impact how they respond to items on an instrument used for an endpoint. If blinding is not possible, please discuss that with FDA. There might be situations where bias can be overcome, for example, if you have very large treatment effects, but this definitely would involve more discussion with FDA. And last but not least, don't forget these important concepts. Firstly, patient input is critical all along the way when you're developing your COA-based endpoint, when you're deciding on the appropriateness of your recall period, when you're determining what effect size is clinically meaningful. Of course, engage early with FDA. We strongly encourage sponsors to introduce their COA endpoints in earlier trials. Gives you an opportunity to see how the COA performs, and will then inform their use in the pivotal trials. And remember, a statistical win on a COA-based endpoint isn't sufficient to establish benefit. The treatment effect really must also be shown to be clinically meaningful. And there'll be more on this topic in the next session. Thanks a lot for your time and attention.
Again, we thank Dr. Joffe for taking the time to provide us with that excellent presentation. Now we will move into the second topic of the webinar, focused on evaluating meaningfulness of treatment benefit. I would like to remind everyone that the recording and slides will be available online following this webinar. We have a Q&A panel at the end of this webinar, but you can feel free to share your questions now by using the Q&A button at the bottom of your screen. Now I will turn it over to Dr. David Reisner from the Division of Clinical Outcome Assessment in the Office of New Drugs in the Center for Drug Evaluation and Research. Dr. Reisner, please turn on your camera and begin your presentation.
Good afternoon, everybody. I'm going to introduce our second topic for the webinar with an overview and a few points to consider. Next slide, please. FDA evaluates how well results of a COA-based endpoint correspond to a clinically meaningful effect of an intervention. We define clinical benefit as a positive effect on how individuals feel, function, or survive, and that definition is taken from the BEST glossary, which is available to the public. Importantly, as you heard from Dr. Jaffe, statistical significance alone does not indicate whether the detected result corresponds to a meaningful treatment effect. Also, as Dr. Gerard discussed earlier, just for clarity, COA-based endpoints are constructed using COA scores. To interpret the meaningfulness of a COA-based endpoint result, we need to know how COA scores relate to patients' experience. This is true whether the endpoint is based on scores generated from a single COA or multiple COAs, as in a multi-component endpoint. Next slide, please. Sometimes COA scores are easier or harder to interpret. For example, we can understand what is meant by the number of times waking up per night. The ordinal metric of none, mild, moderate, severe is also familiar to patients and other reporters and relatively easier understood. In contrast, a metric sensed as a 0 to 10 point numeric rating scale is more difficult to directly interpret. Next slide, please. The need for empirical evidence to interpret the meaningfulness of scores may range from may not be necessary to recommend additional evidence. Next slide, please. In general, begin by reviewing existing evidence to support interpretability of COA scores used to construct the COA endpoint. If the existing evidence is not sufficient, conduct one or more empirical studies to support interpretability. Empirical approaches will generate a range of plausible thresholds, and sponsors should pre-specify a range of thresholds to be used to interpret treatment effects in registration trials. Empirical studies, for instance, may include a psychometric analysis plan to develop evidence from longitudinal data available in a phase 2 study, or to guide the analysis of a parallel COA-focused study. And just stepping back, as you hear more details about specific methods, it's important to remember that if you have a COA that is fit for purpose, you should consider, is the meaning, i.e. what you're trying to measure, preserved in the COA-based endpoint? And that can guide your approach in general. Next slide, please. At this time, I'd like to introduce the next speaker, Dr. Fraser Bosell.
Thank you, David. My name is Fraser Bosell. I'm a psychometrician with the Patient Science and Engagement team at the Center for Devices and Radiological Health. Next slide, please. So as you know, this is a challenging topic. So we've presented some considerations here to keep in mind as I go through my slides today and as you review the draft guidance. So keep in mind that no single method works in all situations. You've got to consider what your goals and your future product submission is going to look like. And also keep in mind that we've made some additions to the terminology to better incorporate some of the ideas that we'll be talking about today. So next slide, please. So to start at the beginning, we're interested in knowing what COA scores tell us about a patient's experience, so that we can define either an endpoint in terms of what's meaningful to patients or interpret the meaningfulness of an estimated treatment effect. Next slide. What's needed is some way to put COA scores in the context of people's lives. Among the different methods that have been developed to do this, we see two broad strategies. The first approach tries to map differences in COA scores to score corresponding differences in patients' experiences. We call this meaningful score difference. Meaningful score differences can be estimated using one or more different empirical methods, which I will discuss shortly. The second approach tries to map COA scores to corresponding patients' experiences. We call this approach meaningful score regions. Meaningful score regions can also be estimated using one or more different empirical methods, and I'm also going to discuss those a little bit later. The estimates of meaningful score differences and meaningful score regions can then be used to inform decisions about defining an endpoint, such as one based on a responder status, or about how to interpret the meaningfulness of an estimated treatment effect. Next slide. Next slide. There we go. Now I'll give examples of methods that you might use to estimate meaningful score differences. These methods all try to connect differences between COA scores to corresponding differences between experiences that patients might have. The goal is not to describe any of these methods in great detail, but rather to explain the logic of how they connect COA scores with patients' experiences or judgments. An anchor-based method uses some external variable for which meaningful differences are directly interpretable or already known. Meaningful differences on an anchor can then be mapped onto differences in terms of the COA scores. In this example, each patient is assessed at baseline and follow-up with a COA and an anchor known as a patient global impression of change. In between baseline and follow-up, the patient might undergo some change in their symptom severity. Using the anchor method, patients categorize their change in symptom severity as either much better, a little bit better, no change, a little bit worse, or much worse. In this case, let's say the patient selected a little bit better. We can then compute a change score for the COA between baseline and follow-up. Collecting these data from many patients allows us to look at the range of COA score change scores for patients who might have judged their experience as getting a little bit better. Statistical methods can then be used to generate estimates of meaningful score differences for these data. Next slide, please. Whereas the last method relied on measuring within patient change, the idioscale judgment method asks patients to consider how different they are from each of several hypothetical patients. So it begins with a patient who has a given score on some multi-item COA. Next, the patient is shown a brief description of the hypothetical patient called a vignette. The vignette is constructed by taking a score value of the COA and computing the most likely item responses for that score. Those likely item responses are used to create a series of statements about the hypothetical patient, such as Omar does not have any difficulty climbing stairs, or Omar sometimes needs assistance getting up from a chair. The patient doing the idioscale judgment task decides whether the hypothetical patient's symptom or function is the same, better, or worse than the patient. The patient makes these judgments for multiple different vignettes that represent scores that are lower or higher than the patient's own score. We can then look at the size of the difference in COA scores between the patient and the hypothetical patients who the patient said were different from themselves to estimate what counts as a meaningfully worse score, a meaningfully better score. Next slide, please. Next, I will discuss several methods that might be used to attempt to estimate meaningful score regions. These methods all try to connect specific COA scores with specific patient experiences or judgments of those experiences. Next slide. The bookmarking method is an approach for finding meaningful score regions when multi-item COA is used. In this method, a patient or groups of patients are shown multiple vignettes that represent hypothetical patients across a range of severity, represented by COA scores. Just like in the idioscale judgment method, these vignettes are created by computing the most likely answer to the items on the measure for a given score, and then making a little story out of those item responses. The patients or group of patients then sort those vignettes into groups representing mild, moderate, or severe health states. By looking at the COA scores that correspond to these divisions, we can get some idea of distinctly meaningful regions of scores. This method takes the less familiar COA score metric and relates it to language that is more intuitive for people to understand. Language of mild, moderate, severe, or none. Next slide, please. An anchor-based method could also be used to inform meaningful score regions. Here, the anchor is the patient global impression of severity, in which each patient rates their symptom severity or functioning as none, mild, moderate, and severe. We could then calculate those score regions. Next slide. When many patients have done this, statistical analyses can be used to understand what regions of COA scores tend to be judged as none, mild, moderate, or severe. This figure shows the distribution of patients' COA scores separately for each response category on the patient global impression of change, or impression of severity, sorry. As with the bookmarking method from the previous slide, the idea here is that we are trying to get a better understanding of how the COA scores relate to a patient's experiences by seeing how patients judge those experiences using more accessible language of none, mild, moderate, and severe. This uses some external variable for which meaningful differences are directly interpretable or already known. Meaningful differences on the anchor can then be mapped to differences in terms of COA scores. In this example, each patient is assessed at baseline and follow-up with the COA score. Oh, excuse me. I believe I've already gone through that. So next slide. So besides the methods I summarized today, Draft Guidance 4 discusses two other methods that can be read, that you can read more about in the guidance. For multi-item measures that form a unidimensional measure, one can select one or more illustrative items to map COA scores to the predicted item responses. Qualitative interviews can also inform what patients regard as distinct experiences related to different COA scores. Next slide. There are different approaches to applying meaning to the score derived from a COA. We talked about meaningful score differences, which include estimates you are familiar with, such as minimally clinically important meaningful differences and responder definitions. The new terminology we introduced is inclusive of this way of conceptualizing meaningful changes or differences. It adds flexibility, as say the minimum may not be a sufficient estimate for all medical products. And other methods and estimates may work better for your particular case. Think about what works best for your goals, your products, and your resources. Remember that all methods have important assumptions and multiple methods are encouraged, and all centers welcome early interactions regarding plan submissions. Finally, we recognize this is a growing and evolving field, and we still need additional research evaluating the methods and comparing the benefits and drawbacks of each. Next slide. Next, Dr. Morel will discuss the use of meaningful score differences and meaningful score regions to either define an endpoint or to help interpret the meaningfulness of an estimated treatment effect. Thank you.
Hello, I'm Dr. Monica Morel. I'm a statistical reviewer on the patient-focused statistical support team in the Office of Biometrics, Division 3 in the Center for Drug Evaluation and Research. Today, I will discuss applying information about meaningful score differences or meaningful score regions to clinical trial data. Next slide. In the previous presentation, Dr. Bosell presented approaches for collecting evidence to support the interpretability of COA-based endpoints. This last session of today's webinar will focus on how to apply such evidence in the form of meaningful score differences and meaningful score regions in the context of clinical trials. Next slide. I will begin by focusing on estimated meaningful score differences and meaningful score regions to define an endpoint. Recall from Dr. Bosell's presentation that we encourage the use of multiple methods, including multiple anchors, to estimate meaningful score differences and regions. This will often result in a range of meaningful score differences and regions. However, when using MSDs or MSRs to define an endpoint, it is important to pre-specify a single score threshold and to provide compelling evidence for that threshold. Next slide. Sometimes sponsors might wish to use meaningful score differences or regions to derive a threshold that is used to define an endpoint, such as a responder endpoint. Recall that a responder endpoint is constructed by dichotomizing ordinal or continuous COA scores. For example, if prior empirical studies indicated that meaningful score difference was 2, then a threshold of 2 or greater might be applied to patients' change from baseline scores to define a responder endpoint or a time to event endpoint. The continuous COA scores would then be dichotomized so that a change from baseline in COA score of 2 or greater would indicate a patient is a responder, and a change from baseline in COA score of less than 2 would indicate the patient is not a responder. Next slide. If instead meaningful score regions approach was used, then an investigator might place a threshold between 2 regions. For example, consider a COA that can generate scores from 0 to 20. A study is conducted with an independent sample of patients using the Patient Global Impression of Severity as an anchor to inform an approximate division of the COA score range into meaningful score regions. Recall that approximate score regions denote groups of COA scores that are thought to be similar to one another and different from other groups of COA scores in terms of the patient's experience of the symptom being measured by the COA. Part of the definition of a responder endpoint might require a patient's COA score to fall in the meaningful score regions of mild or none. Patient scores would then be dichotomized so that patients who are in the score regions of mild or none are classified as responders, and patients who are in the score regions of moderate or severe are classified as non-responders. Next slide. When using meaningful score differences or meaningful score regions to define an endpoint, 2 important considerations should be kept in mind. First, before deciding to use a COA score threshold to construct a responder endpoint, review the special considerations and concerns about responder endpoints discussed earlier by Dr. Gerard. Second, when a COA score threshold is used to define an endpoint, sponsors should pre-specify and provide justification for a single threshold. This is because patients get to have only one value for an endpoint. Next slide. The remainder of this presentation will focus on taking the results from studies of meaningful score differences and using them to help interpret the meaningfulness of an estimated treatment effect. Specifically, we will be discussing the interpretation of treatment effects on an endpoint defined as COA score or change from baseline score at some fixed follow-up time point. There are many considerations when interpreting the meaningfulness of an estimated treatment effect, and we won't have time to cover them all today. Instead, I will focus on a few key ideas and on elaborating some of the foundational concepts that motivate the recommendations in Guidance 4. This will include some material not explicitly found in the guidance. Next slide. After the finding of a statistically significant treatment effect on a COA-based endpoint, we need to understand, is that treatment effect meaningful to patients? How the treatment effect is estimated will depend on the endpoint and the trial design. For our discussion, we will assume an endpoint based on a COA score or a change from baseline score at a fixed time point and a parallel groups design. In parallel groups designs, the treatment effect is found by comparing study groups. How we examine the meaningfulness of this treatment effect depends on how we look at the group comparison. Next slide. The different ways of looking at the group comparison can be illustrated using empirical cumulative distribution function curves for each treatment group. We will come back to this specific example shown on the slide later. First, I will introduce two ways we can use ECDF curves to examine the meaningfulness of an estimated treatment effect by comparing the treatment groups. Next slide. One approach we'll discuss involves looking at group comparisons in terms of the expected difference in the probability of exceeding one or more score thresholds. That is, examining the vertical gap between the two groups. This approach allows us to answer the question, how much more likely is the average patient to experience a meaningful improvement in how they feel if given the drug rather than the placebo? Next slide. The other approach we'll discuss involves looking at group comparisons in terms of expected differences in scores or in change from baseline scores. That is, examining the horizontal gap between the two groups. This approach allows us to answer the question, how much better is the average patient likely to feel if they receive the drug rather than the placebo? In the next two slides, I will give a quick overview of these two approaches. The rest of the presentation will then focus on a more detailed illustrative example using both approaches. Next slide. When we examine the meaningfulness of an estimated treatment effect by looking at differences across treatment groups and the probability that the average patient score exceeds some threshold or thresholds, we are really focusing on the vertical gap between the two groups' ECDF curves. Under this approach, estimated meaningful score differences or regions are used to inform thresholds. Then, the probability of exceeding a given threshold across the treatment group is compared. Finally, a determination is made about whether the magnitude of difference in those probabilities is clinically meaningful to patients. Next slide. When we examine the meaningfulness of the treatment effect by looking at differences across treatment groups in expected scores, we're examining the horizontal gap between the two groups' ECDF curves. In this case, we're looking not at the probability or likelihood of having a certain change in the endpoint or scores, like on the last slide. Instead, on this slide, we're going to talk about looking across the X or the bottom axis of the graph, across the different changes in the endpoint or scores. As the average horizontal gap is equal to the difference between the means of the two score distributions, the average horizontal gap corresponds to the overall treatment effect estimated from, for example, an ANCOVA model. The treatment effect obtained from an ANCOVA model can therefore be viewed in conjunction with the range of thresholds informed by MSDs or MSRs to aid in the determination of whether the estimated treatment effect is meaningful to patients. Next slide. I will now walk through an illustrative example that uses meaningful score differences. We will discuss how the meaningfulness of the treatment effect can be examined using both approaches just described. That is, by looking at the expected difference in the probability of exceeding one or more score thresholds across treatment groups, and by looking at the expected difference in scores or change from baseline scores across treatment groups. For this example, we will assume anchor-based analyses were conducted to estimate a range of meaningful score differences. Next slide. Here are the details of our hypothetical example. Our data are from a randomized clinical trial comparing a drug and placebo. The trial used parallel groups design and had a sample size of 250. The COA-based endpoint of interest is a change in ABC symptom index score from baseline to week 24. The ABC symptom index was assessed at baseline and at 24 weeks post-randomization. Scores on the COA can range from 0 to 40, and higher scores indicate greater symptom severity. The primary efficacy analysis was a comparison of study group mean change from baseline scores using analysis of covariance with baseline ABC symptom scores as a covariate. Next slide. A meaningful score derivation study was conducted prior to the registrational clinical trial. A patient global impression of severity scale was used as an anchor measure. Using the data from this separate study, a range of MSDs was estimated, in part by examining the distribution of the change from baseline in ABC symptom index scores and the sample corresponding to a change in PGIS that was determined to be meaningful to patients in qualitative interviews. Here, that's a 1-point improvement on the PGIS. The range of meaningful score differences was estimated to be -7.5 to -8.5. Next slide. This slide shows the results of the primary efficacy analysis for our hypothetical randomized clinical trial. The estimated treatment effect, which is statistically significant, is -8.3. This estimate can be interpreted as a causal effect of treatment for the typical patient in the trial. Note that this interpretation is possible because the treatment effect estimated from this linear model is a conditional treatment effect, a point that is further explained in FDA draft guidance for industry adjusting for covariates in randomized clinical trials for drugs and biological products. The results of the efficacy analysis for our COA-based endpoint was statistically significant. Now we need to understand if this estimated treatment effect is meaningful to patients. Next slide. To aid in this, we plot the ECDF curves for change from baseline to week 24 in ABC symptom index scores by treatment group. As we discussed earlier in this presentation, how we examine the meaningfulness of the treatment effect will depend on how we look at the group comparison. Next slide. We begin by looking at the expected difference in the probability of exceeding the MSD thresholds, which were derived using anchor-based methods. This will allow us to answer the question, how much more likely is the average patient to experience a meaningful improvement in their ABC symptoms if given the drug rather than the placebo? Next slide. Recall that under this approach, estimated MSDs are used to inform thresholds, here -7.5 to -8.5. Then the probability of exceeding these thresholds across the treatment groups is compared. This difference corresponds to the vertical gap between the groups. Finally, a determination is made about whether the magnitude of difference in those probabilities is clinically meaningful to patients. Next slide. As recommended by Ravicki and colleagues in 2007, it's helpful to directly plot this vertical gap as a function of the endpoint values. Here our x-axis is the same, but the y-axis is now the distance that was between the two lines, the treatment and control arms, on the last slide. Then we can inspect the difference between the groups in the probability of exceeding a meaningful score difference. Next slide. For example, here we overlay the MSDs of -7.5 to -8.5, which corresponds to a difference in the probability of decreases greater than those MSDs of 18 to 20.5%. This range of percentages answers the question, how much more likely is the average patient to experience a meaningful improvement in their ABC symptoms if given the drug rather than the placebo? Next slide. It's important to note, however, that as reported in a recent paper by FDA authors, the size of the difference at any score threshold is dependent not only upon the separation between the group means, but also on the amount of within-group variance. The smaller the variance, the larger we expect the difference in probability of exceeding a threshold to be. In other words, those bigger differences in probabilities may be driven by within-group variance. Next slide. We will now examine the meaningfulness of the treatment effect by looking at the expected difference in change from baseline to week 24 in ABC symptom index scores across treatment groups. This will allow us to answer the question, how much better is the average patient's ABC symptoms likely to be if they receive the drug rather than the placebo? Next slide. Recall that the overall estimate of treatment effect, which is based on comparing group means, corresponds to the average horizontal gap between those ECDF curves. To see whether the difference in group means might be driven by particular areas of the distribution, we can examine the consistency of the horizontal gap as we move up the y-axis. This can be challenging to perceive, so it's useful to directly plot the horizontal gap as a function of the cumulative percentage. Next slide. Directly plotting the horizontal gap allows us to more easily examine whether the gap is consistent. This figure shows the horizontal gap in our hypothetical example is relatively consistent in the parts of the graph of most interest to us, which supports the use of the difference in group means as an estimate of the treatment effect. Next slide. Here's a display of the average horizontal gap for the ECDF plot we just saw. It is the estimated treatment effect from the ANCOVA model, adjusting for baseline ABC symptom index scores. The treatment effect represents the expected difference between drug and placebo in change from baseline ABC symptom index scores for the average patient. In other words, it answers the question, how much better are the average patient's ABC symptoms likely to be if they receive the drug rather than the placebo? Next slide. To help put the treatment effect in context, we can overlay the estimates of the meaningful score difference thresholds on the estimate of the treatment effect. Next slide. MSD reflect thresholds for meaningfulness that are likely to vary from patient to patient. So there's no single precise threshold. It is actually more helpful to have multiple estimates of MSDs using different methods to help create a richer context for interpretation and to aid in the determination of whether the estimated treatment effect is meaningful to patients. It is important to note that viewing the estimated treatment effect in the context of MSDs is not like a second level of hypothesis testing, where we see whether the lower bound of the 95% confidence interval exceeds some threshold. Next slide. Now that we've walked through two approaches to examining the meaningfulness of a treatment effect, which depend on how we look at the group comparison, let's take a step back and review what we've done in this hypothetical example. To estimate meaningful score differences, we conducted a study prior to the clinical trial that used an anchor-based method with the Patient Global Impression of Severity scale. We looked at the distribution of ABC symptom index change scores for the groups of patients whose global impression of severity ratings improved by one category, which patients told us in qualitative interviews was meaningful to them. From looking at these data, we decided on a likely range of meaningful score differences. That is, the expected difference in scores perceived as meaningful by the average patient. This range was -7.5 to -8.5. Next slide. Then, in the hypothetical clinical trial, we compared distributions from groups of patients, in this case between patients who were receiving drug and placebo, using two approaches. First, we looked at the expected difference across treatment groups in the probability of exceeding the MSD thresholds of 7.5 to 8.5. We found that this difference was between 18 and 20.5%. Second, we looked at the expected difference across treatment groups in the change from baseline to week 24 ABC symptom index scores. To put the treatment effect estimated from the ANCOVA model into context, we overlaid the thresholds derived from the meaningful score differences on the treatment effect estimate and corresponding 95% confidence interval, and determined the magnitude of the treatment effect estimated for the average patient is in the range of what the typical patient would consider meaningful. Next slide. So in this session, we have discussed some of the foundational concepts that motivate the recommendations in Guidance 4. We discussed how the meaningfulness of the treatment effect can be examined from two perspectives, examining expected difference in probability of exceeding one or more score thresholds, and by examining expected differences in scores or change from baseline scores. With both approaches, MSDs and MSRs can play a role in helping to interpret whether the treatment effect is meaningful to patients. Next slide. To conclude, I'd like to make a few summary remarks. And although we only discussed meaningful score regions in the context of defining an endpoint at the beginning of this presentation, and not in the context of interpreting treatment effects specifically. All of these comments will apply to meaningful score regions as well as meaningful score differences. Using MSDs and MSRs to interpret the meaningfulness of a treatment effect is not meant to be like using an algorithm to produce a yes or no answer about meaningfulness. Instead, it is about creating a richer context in which to view the estimates of treatment effect. Determining whether a medical product produces an effect that is meaningful to patients involves careful consideration of multiple sources of information. This could include findings from multiple methodologies or anchors to derive MSDs or MSRs, treatment effects from other endpoints, pre-specified sensitivity analyses to supplement the main trial analyses of the COA-based endpoint, analyses to examine heterogeneity of treatment effect, and graphical as well as exploratory analyses to examine analytic assumptions or illustrate findings in alternative ways. Sponsors should pre-specify the methods used to interpret COA-based treatment effects and to convey the uncertainty around the estimates of MSDs or MSRs. Next slide. Much of draft guidance 4 is about the math and submissions to FDA. It focuses on analyzing and interpreting trial data, but this is of course only one piece of the patient-focused drug development puzzle. This guidance series as a whole is at an intersection that many FDA review teams, sponsors, and patient groups are quite familiar with. To make decisions, we simultaneously are using information from qualitative and quantitative studies, moving from talking to individuals and groups, creating and testing COA measures, using those measures in studies and trials, and analyzing the data we receive. Next slide. All of this type of information is used by FDA, sponsors, and patient groups to create a rich context in which to understand the effect a medical product has on a patient's life. The desire to understand the patient experience and to find clarity using the data in a rigorous and systematic manner to support regulatory decisions is expressed every day by our leadership and reviewers across our centers. As we move forward with PFDD, we want to continue to do so rigorously and with maximal flexibility. These guidances are part of that mission. Thank you. I will now turn the webinar over to Shannon Sparkling for Q&A.
Thank you, Dr. Morell. Now we will transition to the Q&A session. At this time, all presenters should please turn on their cameras. All right, great. So the first question that we received from the webinar is for Dr. Luo, and the question says, it is often difficult for sponsors to understand how different divisions within FDA interpret and use the guidance. To what extent do the views on the guidance shared by CDER representatives reflect those held by CBER?
Okay, that's a great question. This is the 3-center guidance. All the medical product offices, CDER, CBER, and CDRH are all issuing the guidance. So the guidance should apply to all 3 medical product centers. Thank you.
Great, thank you. The next question is for all of the panelists, and the question is, how does someone request a meeting with FDA to propose a new COA? So perhaps Dr. Bosell, we can start with you.
Yes, thank you. So for CDRH, there's actually a couple of pathways. The main pathway is the Q submission program, which is laid out in the Q submission guidance. There are several types of Q submissions to fit your needs, and you can also request that the patient science and engagement team be involved when the Q sub is submitted. Alternatively, you can also look into the medical device development tool program, which is a pathway to qualify your tool outside of a specific medical device submission. And more information on that is available on our website and also through guidance.
Great, thank you. Now I'll turn to our CDER and CBER colleagues.
Sure.
That question.
Oh, sure. And I can start. So I'm Dr. Laura Lee Johnson in CDER. And if a medical product has been identified, and my CBER colleagues can correct me if I get any of this wrong, for CBER and for CDER, We have what are called type A, type B, type C, and also type D meetings. And so some of these are pre-IND meetings, some of these are IND meetings. And so it's important if you have a product that's within one of those programs, so you've already approached the FDA or you are now approaching CDER or CBER, then you can come in through one of those meeting types. And so we get quite a few of these as written response only type C meetings and also these newer type D meetings. Now, if you don't have a product yet, we also have, and CBER actually had this first, but under PDUFA 7, CDER also has it, these new interact meetings, and that is a potential option. And you can go onto our websites and learn more about those. For rare diseases, we have a new pilot also under PDUFA 7, and that's the Rare Disease Endpoint Advancement or RDEA pilot program. And that is just now starting. In fact, we have our first virtual workshop June 7th and 8th. So if you're interested, you can go and look that up. Now, Dr. Reasoner, I don't know if you want to talk a little bit about our CPIM program and qualification, maybe?
Yes, thank you. I can comment briefly. So with respect to the CPIM program, it's important to note that the program is intended for situations where there's not a specific medical product development program under discussion, but more broadly measurement issues that may be impacting, for instance, the development of an individual instrument or even a whole field of measurement. And you're seeking broad regulatory input. It's often a very senior and diverse group of participants, so it can be a valuable experience. And the other consideration might be whether the clinical outcome assessment, the COA qualification program, could be a vehicle for receiving regulatory guidance. We have several DDT programs, one of which is the COA one, which is relevant to today's discussion. So there's material on the scope of the program, the objectives, the criteria for acceptance, et cetera, that's available on the FDA website. Thank you.
Great, thank you all for providing that helpful information. The next question we have is for Dr. Morell. The question asks, would you weigh the components equally when constructing a multi-component endpoint, or do you need to demonstrate the weighing of the component with qualitative evidence or quantitative evidence at baseline of overall disease severity?
Sure, thanks for that question. So sponsors should be explicit about how each component is weighed and provide a justification for those weights using compelling evidence. That could include qualitative and or quantitative evidence. The weighing itself does not need to be equal.
Great, thank you. The next question we have is for Dr. Johnson. The question asks, does the agency have statistical preferences for treating missing COA data due to technical problems with the ePRO devices?
Well, many of y'all may be familiar with our digital health technologies guidance as well. And I think there's a lot of different information available thinking about these. Now, there's really no single preferred method that applies to all situations because there can be a lot of different problems and a lot of impacts of those problems. So sponsors should propose and justify the approach to handling the missing data. And we always recommend coming and discussing with FDA prior to finalizing that statistical analysis plan and definitely prior to unblinding. We recommend testing also your ePRO devices prior to initiating, especially those late phase trials, in order to proactively identify and address the potential problems that could be coming up.
Great, thank you very much. The next question is for Dr. Bosell. The question asks, what studies are within the scope of this guidance? It is mostly geared towards P3 pivotal registrational, or is FDA expecting to engage with sponsors on COA topics for P2 studies if they might be used as supportive data for effectiveness?
Yeah, great. Thanks for the question. So these guidances are appropriate for all types of studies. We think it applies throughout the total product life cycle. And we encourage, whenever possible, to engagement far in advance of the pivotal and registrational trials.
Great, thank you. This next question is for Dr. Johnson and Dr. Bosell, so perhaps we can start with Dr. Johnson. The question asks, where/how should evidence to support the interpretability of the COA be provided to FDA? For example, CSR, PRO dossier, protocol, SAP. How would that change depending on whether the data are existing or derived empirically?
There's a lot in this question, so I'll try to get a good start on it. So where you put the evidence and how you actually provide it to FDA, we discussed that a bit in the last section of draft guidance 4. And a lot of it is going to depend on kind of where you are. So for example, evidence can be submitted at the IND stage, and that might be in a PRO dossier that comes in at the IND stage. Sometimes we see PRO dossiers, maybe we would see it also if you're coming in for that marketing application. So for us in CDER and CBER, it's usually an NDA or BLA. And that's an important part, but also the protocols and the statistical analysis plans, those come in prior to finishing the drug development program and coming in on that marketing application. So that might come in during the IND. So a lot of times that location, it's the meeting package isn't going to change if the data are existing or if it's a program specific data. So if you're going to be developing additional data within your program, kind of where it comes in to us, kind of the data is the data, the information is the information. So if you have evidence that, for example, is going to support the interpretability of a COA, you may really want to start talking to us about it early. So if you have a traditional 3-phase program, you may want to come in at the end of phase 2 or even earlier to have that discussion, during which the time you're talking about the proposed assessments and endpoints and analysis plans, et cetera, because it can really tie into a lot of the study designs, especially for those confirmatory trials. But we need that additional information. I'll stop here, pause here briefly. I don't know, Dr. Bosell, if you want to add in, I know you all have different types of meetings and setups at CDRH, so you may want to say a bit more.
Yeah, I think we have similar thoughts on this. I just have to translate it for my audience. So I'm going to sound a little bit like a broken record because for a device, the Q submission program is really a great opportunity to come in and present this information to us and get feedback on specific questions. Generally speaking, we would prefer you come in through a Q sub outside of the investigational device exemption submission. You can bring it in when you're submitting the statistical analysis plan prior to your either pre-market approval or de novo or whatever approval or clearance submission you're going to be submitting to us. And so we really think that's the best opportunity to come in and talk to us is through that Q submission process, and the timing is always the earlier the better.
Great, thank you both. This next question is for Dr. Morell. The question asks, are anchor-based approaches still recommended as the best way to obtain data to inform interpretation of treatment effects?
So FDA continues to view anchor-based analyses as a useful method for understanding what types of COA score differences, including changes, are meaningful to patients. But it's important to emphasize the critical assumptions that are made by anchor-based approaches. Guidance 4 elaborates on the need to explore these assumptions. And Guidance 4 also discusses other methods that can be used in addition to or instead of anchor-based approaches. In general, sponsors should consider and pre-specify the approaches that are most appropriate for their particular situation that can help put COA score differences in the context of patients' lives. FDA does not recommend any single approach and encourages multiple approaches, which could even be multiple anchors to be used to create a context for understanding treatment effects.
Great, thank you. We have one more question for Dr. Morell. The question asks, what recommendation does the agency have for estimating weights for a multi-component endpoint?
So there's no one recommended approach for determining weights for a multi-component endpoint. The approach should be informed by patients and or caregiver perspectives about the importance of the individual components themselves. This is often informed by a summary of item and score performance using one quantitative method or another. Thank you.
Thank you. This next question is for Dr. Johnson. The question asks, in rare diseases where patient numbers are small and there is significant urgency in meeting an unmet medical need, is there flexibility in the level and amount of evidence required to support a COA-based endpoints use?
Sure. So flexibility should be discussed with the review divisions. So, and I think Dr. Gerard tried to talk about this some. I think in the guidance, we're trying to describe a variety of approaches and potential flexibilities. In the rare disease space, we still have to get to substantial evidence of effectiveness, and that's much broader, and there's a whole other guidance for that. But it is important to make sure that we are rigorous in what we're doing. And that's the key. But again, I think we have a lot of good examples where we've been able to work with sponsors and patient groups to get to where we need to be to make sure that we have a drug that is safe and effective for patients on the market. And that's true for all medical products, not just drugs.
Great, thank you. Now we'll go to our last question, which is for Dr. Reasoner. The question asks, what is a type D meeting?
Sure. A type C meeting? Well, a type C meeting can be used for many purposes. And in the COA space, I think what I would suggest is that it can be the basis of having a COA focused meeting. I mean, oftentimes, particularly if you're modifying an instrument or even developing a new instrument, you may have a series of questions that you would like to pose in a meeting package. And this type C meeting is a mechanism by which you can share those questions and your background information with not only the review division, but the Division of Clinical Outcome Assessment. And I'd encourage you to include all the necessary background material that you would expect is needed to answer the questions you're submitting. Thank you.
Shannon, did you say type D or type C?
I did say type D.
Okay. Yeah, so I guess the difference between a type C and type D meeting is the type D meetings are focused on a very narrow set of issues. So it's usually no more than, say, 2 very focused topics. So that's, and you can look online at our meeting guidances and it'll give you more details about those. Just a little add on there.
Thank you. Sorry, Shannon, I misheard.
No worries. We received some helpful information about type C meetings as well.
We do a lot of type C meetings that are really focused.
All right, well, thank you all. That concludes our Q&A session. Thank you all in the audience for submitting your questions, and thank you to our presenters for their contributions. Next slide, please. Another reminder that Guidance 4 has an associated public docket. The docket is open to the public and will close on July 5th, 2023. To submit a comment, please access the docket using the link on this slide and click comment. If possible, please submit your comments in a Word document format. When applicable, it is helpful to specify line numbers and propose text revisions for comments and propose changes to the guidance. In addition to any proposed changes, it is also helpful for us to hear what you like about the guidance, so we can consider both types of feedback when determining what to revise or remove. I would like to remind everyone that the recording and the slides will be available online following this webinar. I would like to thank everyone involved in the planning of this webinar, and I would also like to thank our presenters for their participation. Have a great afternoon.